Hi. So, in Advanced Bioinformatics lecture, uh, we will start with the overview of bioinformatics, which is an interdisciplinary field that combines major principles from the core concepts, the core fields like biology, computer science, mathematics, and statistics. And why we do this? Because already in high throughput technologies like sequencing, we are generating a huge amount of DNA and protein sequencing data. And all these data sets and these large scale biological data sets, which we are referring to, uh, are growing rapidly. And in order to collect and store and analyze and interpret with these biological data, it is essential to uh, combine all these fields. So, by combining all these in major fields like biology and computer science and statistics, a main goal revolves around managing and organizing this data, and then identifying patterns and relationships in these sequences in order to identify mutations and disease relationships, and then understanding gene structure, function, and regulation, how these regulations and gene structures are being varied by the variation of uh, the changes in sequences, and then predicting protein structure and function, how the translation and then ultimate translation ultimately is actually controlling the protein structure and function. Supporting system biology, genomics, transcriptomics, proteomics, and metabolomics is another another key goal of bioinformatics. By all these, managing and identification of biological sequences and gene structure understanding, all these leads to a major role of bioinformatics in modern biotechnology, in personalized medication, in evolutionary biology study, in agriculture, in revolutionary medicine, involve enabling discoveries that would not be possible through experimental biology alone. It's always better to have at least some calculations before actually going to the lab and performing some experiments. So this information is the core, it provides the basic and core base to the experimental data. So one of the most important things in bioinformatics is data management. Data management involves two major things: data retrieval and data analysis. This whole large-scale genome projects, currently we are dealing with larger genome projects like microbes, animals, and human genome projects, generating massive amount of data. So we need software tools, specialized software tools to efficiently retrieve and store and analyze this data. So data retrieval in bioinformatics majorly involves systematic collection. It's not random. It is actually systematic, from where you actually get the data, collect the data from public databases and repositories that already store it after verification. So main purpose of data retrieval is to access existing nucleic acids and proteins and amino acids data and their sequences, and then structural information which is already being generated worldwide. And what is the main purpose of accessing this already existing data is to avoid any duplication of experimentation, to avoid wastage of time and resources, and to provide a reliable foundation for comparative studies like whatever you are doing and whatever already is being done, and computational studies to compute for making basic generation of information for getting experimental data with previously known biological information, integration of experimental data. Data retrieval ensures that researchers work with accurate, standardized, and up-to-date data sets. So next, after retrieval, data analysis is a major step which involves computational and various statistical methods to identify genes and regulatory elements and functional motifs, and to compare sequences to determine if they are just similar or they actually in terms of similarity, or they actually share some evolutionary history in terms of homology and have their evolutionary relationships, and then predict protein structures and their functions, and predict some ancestral history using protein structure and function, and then analyzing gene expression and biological pathways, detect these mutations, and the result of these mutations in form of variants and disease associated changes. Purpose of analysis is to convert raw biological data into interpretable knowledge. So together this data retrieval and analysis is the core of bioinformatics, and where data retrieval provides the information, data analysis actually provides the understanding. So workflow, as we have discussed, is majorly first of all generation, and then acquisition, and then pre-processing, uh, by quality controlling, trimming and filtering errors and low quality data, and then analysis of using computational tools for sequence alignment, genome assembly, and then interpretation of these results, such as gene function, pathways, or disease relevance. And then we have biological insight and hypothesis generation, which is the final outcome. Various software tools which are used for data retrieval and analysis are actually grouped into one. Full databases and tool set is for data retrieval to store and provide access to biological data, and the other is data analysis tool, which is actually to analyze and interpret biological data, which involves statistical and computational tools. So that's all for the first lecture. Thank you.